All right, so let's get into the nitty-gritty of how photosynthesis works. First thing that happens is the sun, the ultimate source of energy on Earth, is going to hit the chloroplast on a leaf. Automatically, any water that is there is going to go through photolysis, which is splitting with light. All right, so it's going to split water, you should have an arrow here, into a molecule of oxygen, protons, and electrons. This provides oxygen for the atmosphere that we breathe. It provides us with protons, which we'll talk about later, and it provides electrons. These electrons that are associated in photosystem 2, by the way, in the antenna complex is going to absorb those photons of light. This absorbed energy is going to move from one chlorophyll to another chlorophyll to another chlorophyll until eventually it falls into the reaction center. In the reaction center, that electron is going to get energized. It's going to move to a higher energy level, farther away from the nucleus. Energized electrons are going to get accepted to a primary electron acceptor, and then it's going to move through the electron transport chain. So here's a picture. The ultimate source of energy on Earth, sunlight, is going to go down. It's going to hit the chlorophyll in the plant. It's going to bounce around in the antenna complex until it hits the reaction center, which is actually going to be a funnel down to that. That's going to boost the energy of an electron to a higher energy level. It's going to be accepted by a primary electron acceptor. This is all happening in a thylakoid of a chloroplast. And this is happening in photosystem 2. Now there is a name to this primary electron acceptor. You don't have to worry about that. But this primary electron acceptor is going to take that electron, it's going to pass it to plastoquinone, and then it's going to pass it to cytochrome complex, and then it's going to pass it to plastocyanin. So this is part of the electron transport chain, where the electron just keeps going to one step, to another step, to another step, to another step. Each step of this electron transport chain is creating ATP by chemiosmosis. Okay, so here is our photon that hit photosystem 2. That creates water that splits from the photolysis. We get our oxygen that's going to be released out of the cell. And then we have protons, which we'll talk about later. And then what happens is this electron that gets excited is going to pass from one molecule, the primary electron acceptor, to plastoquinone, to cytochrome complex, to plastocyanin, and so on. We didn't get any farther than that. So we made a little bit of ATP, but now we have an electron that doesn't have any more energy. And energy got transferred to ATP. So this tired electron is now going to need more energy. So it travels to photosystem 1, and it's going to receive more sunlight. So the sunlight hits photosystem 1 now. It gets re-energized, and it gets accepted again by a primary electron acceptor. The primary electron acceptor is going to pass the electron down the electron transport chain to 6 different electron acceptors, with the final electron acceptor being ferredoxin. Ferredoxin is going to transfer that electron's energy to NADP+, which is going to create NADPH, which is our energy molecule that we want. And then that NADPH is going to be released into the stroma. All right, so let's do this again. Sunlight hits the chloroplast. It goes to the antenna complex, to the chlorophyll. Electron gets excited. It goes to a primary electron acceptor, to plastoquinone, the cytochrome complex, to plastocyanin. Each step of this is creating ATP by chemiosmosis. And these electrons were made from the splitting of water, photolysis right here. All right, so then our tired electron travels to photosystem 1, where it goes to another chlorophyll. Another boost of sunlight hits the chlorophyll. It gets bounced around until it hits the reaction center. The electron gets boosted with energy again. It goes to a primary acceptor. And we're going 1, 2, 3, 4, 5, 6 more times with the sixth electron acceptor being ferredoxin. And that energy is going to create, with the hydrogens that we've produced over here from the splitting of water, to make NADPH, which is our energy molecule. So at this step, we have made ATP and NADPH. So from the activity of the electron transport chain, we're getting a lot of protons created from the splitting of water. And these protons kind of gather on the thylakoid membrane, and they get built up in there. So due to the high amount of protons out here, what happens is they end up getting pumped through this protein called ATP synthase, and they get pumped through this, and that drives the energy. This is called chemiosmosis, and this drives the energy to add inorganic phosphate to ADP to create ATP. So this picture shows the electron transport chain in chemiosmosis. So you can see the electron travels in the orange arrow, and then you see the protons that are being gathered here in the middle or the lumen of this thylakoid. And so all these protons are gathered. There's a high concentration of protons here. There's not a lot of, down here. So the protons get pumped through this protein called ATP synthase, and that drives this reaction right here to make ATP. Okay, so basically it's now what? Now what happens? All the light-dependent phase did was transfer the energy from the sun into ATP and NADPH. We haven't made any glucose yet, and we haven't even talked about carbon dioxide. So obviously that stage is next, and that stage is called the carbon fixation stage. All right, so the second part of the reaction of photosynthesis are called the dark reactions. It's so called because they don't need light. It doesn't necessarily happen in the dark, but they don't need light for it to happen. All of this occurs in the stroma of the chloroplast, not in the thylakoid. And basically we are fixing carbon to make our carbohydrates. These reactions are called the Kelvin cycle or the Kelvin-Benson cycle. Well, the guy's name who discovered the Kelvin cycle is called Melvin Kelvin. He discovered it and received the Nobel Peace Prize in 1961. The Kelvin cycle means fixing carbon or putting together carbons. Here's your summary equation for the dark reactions. We're going to use our energy that we made. We're going to use that NADPH that we just made. We're going to use our ATP that we just made. And we're going to finally use our carbon dioxide. And we're going to make glucose. And we're breaking down NADPH into NADP+ and our inorganic phosphates. And we're using the energy of ATP. So we're breaking that down into ADP. And of course, we're making a little bit of water. There are 3 phases of the Kelvin cycle: carbon dioxide uptake, carbon reduction, and RuBP regeneration. First stage is called the carbon dioxide uptake. Carbon dioxide is going to react with a 5-carbon molecule called RuBP. It stands for ribulose biphosphate. This reaction is catalyzed by the enzyme Rubisco. Rubisco actually has a long name to it, ribulose biphosphate carboxylase. So instead of saying that long, ridiculous name, we say Rubisco. This enzyme is in huge amounts in the chloroplast, so it's always there. So that Rubisco allows us to react a 5-carbon molecule called RuBP with the carbon dioxide. So 5 carbons plus carbon dioxide makes a 6-carbon molecule. The product that is formed is a 6-carbon unstable molecule, which immediately is going to break down into 2 molecules of PGA, which stands for phosphoglycerate. Each of these PGA molecules have 3 carbons each. So this diagram is just showing you the first part of the carbon dioxide uptake phase, where we have carbon dioxide, which is going to be catalyzed by ribulose biphosphate, added to that, and then that product splits into PGA, which we have 2 of them. Second stage is called carbon reduction. Our PGA is going to be converted to G3P, which is also nicknamed PGAL. This reaction requires a lot of ATP and NADPH, which we made already in the light-dependent reactions. G3P is then converted to sugars like glucose and other carbohydrates. Not all G3Ps can make sugar, and some of them will go on to phase 3. So here is phase 2, the carbon reduction phase, and you can see here's our PGA. We're going to use ATP and we're going to use our NADPH energy to turn PGA into G3P. The last stage is the regeneration of RuBP, because this is a cycle. We need to make more of that. So G3P can convert to RP. RP will then convert to RuBP using a little bit more energy in the form of ATP. Now RuBP is available again to join with carbon dioxide. So here for stage 3, which is RuBP generation phase, it does show that G3P can go ahead and make glucose, or it will go and turn into RP and then back to RuBP. So there is a separate little pathway there. But otherwise, the whole cycle is making that RuP again so that it can combine with carbon dioxide and start stage 1 again. Here's a picture I found online of all 3 stages. Carbon dioxide, catalyzed with Rubisco, adds to RuBP to make PGA. PGA is broken down using energy to turn into G3P. G3P can make glucose, or it can be converted to RP and then back to RuBP. This is called the Calvin cycle. The Calvin cycle is called the C3 pathway. It's named this because G3P has 3 carbons. This pathway is very inefficient. Less than 1% of the light energy that we got from the sun reaches the chloroplast is found in glucose. It's very, very inefficient, and it's all the enzyme Rubisco's fault. Rubisco is horrible at its job. When there is a lot of light and there's a lot of high temperature, photosynthesis is very active. It's going on all the time. Because of the high light and the temperature, water is easily lost. So the leaf stomatas close. Stomatas are little pores on the underside of the leaf. They close to protect the plant from losing too much water. So what happens is we can't get the oxygen out. Oxygen builds up. And what the problem is that Rubisco really likes oxygen. Rubisco can bond with oxygen as well as carbon dioxide. And so it has an affinity, which means love, for oxygen and carbon dioxide. And so if oxygen is around, it'll bond to it when oxygen is abundant, which means it's there a lot. And so if that happens, G3P will not be produced. This result is called photorespiration. In the presence of oxygen and light, carbon dioxide or carbohydrates are oxidized and therefore no glucose is produced. Instead, carbon dioxide and water is produced. So in C3 plants, 50% of the glucose made might be reoxidized back into carbon dioxide due to this problem. So photorespiration reduces the efficiency of C3 plants because Rubisco can bind to oxygen as much as it binds to carbon dioxide. However, we do have certain plants, they're called C4 plants, where there's a lot of carbon dioxide, so we don't see that cheating, quote unquote, that Rubisco's doing. So photorespiration is going to be limited in these types of plants. So the way that we can work around this problem of photorespiration is the C4 pathway, or the CAM. And the C4 pathway, it's a physical separation for Rubisco away from oxygen, because Rubisco likes oxygen so much, we need to separate them completely. So they use different parts of the plant to avoid that from happening. And the C4 pathway occurs in plants like crabgrass, corn, sugarcane, for example. The other workaround to avoid photorespiration is the CAM. CAM plants carry out in their fixation separated in time, where there's not a lot of high temperatures and high oxygen occurring. And this occurs in a lot of desert plants. In the C4 pathway, there is a different enzyme. It's called PEP carboxylase that converts the 3-carbon compound to a 4-carbon compound, and that's why it is called the C4 pathway. We also have a difference in location. In the C3 pathways, we're using the mesophyll cells, and C4 pathways, they're doing a lot of the reactions in the bundle sheath cells as well. You don't have to know all these details, but you should know that there's a different location of where these things are occurring. There's a different enzyme involved, and so photorespiration just doesn't take place as much as the C3 pathway. This enzyme that we're using instead, called PEP carboxylase, it loves carbon dioxide like it's supposed to, not like Rubisco. It likes carbon dioxide more than oxygen. So that enzyme is faster at getting the carbon dioxide to bond to the RuBP. So here's a comparison for C3 and C4. C3 have Rubisco in the mesophyll cells, and C4 has the PEP carboxylase enzyme in their mesophyll cells. They both have Rubisco. The bundle sheath cells in C3 have very few chloroplasts in them and no Rubisco. And the C4 does have Rubisco, but only in the bundle sheath cells. The bundle sheath cells for the C4 are close to the mesophyll cells, which allows carbon dioxide to transfer between them. Here's a diagram showing again the difference in the C3 versus the C4 anatomy of a leaf. So again, C3 has extensive photorespiration occurring. C4 has minimal photorespiration occurring. C3 and C4 both do the Calvin cycle. What's accepting the carbon dioxide is different. It's a different enzyme. In C3, it's RuBP, and it's Rubisco. And in C4, it's a molecule called PEP, and the enzymes, it's using PEP carboxylase and the Rubisco, so it has both enzymes. In C3, the final product, or the first product of fixation is a 3-carbon compound called PGA, and in the C4 pathway, it's a 4-carbon compound called oxaloacetate. In C3, we have a moderate love for carbon dioxide occurring, and in C4, we have a higher love of carbon dioxide, so the bonding is more efficient and better and faster. In C3, it's occurring in the mesophyll cells only, and in C4, it's occurring in the mesophyll and bundle sheath cells. For the last one, the CAM pathway, it's seen in desert plants, and the separation is done by time instead of by location. The enzyme, they use PEP carboxylase enzyme, and it only works at night. It only works when the stomatas are open. They're closed during the day in desert plants because it's hot. Oxaloacetate is made, malate is derived from it, it's stored in the vacuole. During the day, carbon dioxide is made from the malate and made available to the Calvin cycle. So it's not as efficient as supporting rapid growth as seen in C4, so it's not as good as C4, but it's a good adaptation for those type of plants. So the last thing is just that Calvin cycle is truly the most important part of photosynthesis because that is what is making the carbohydrate, making the glucose, and that's what's the most important, no matter what kind of plant it is. It's the Calvin cycle. That's the most important thing.